students, welcome back to another YouTube tutorial from Naeem Bukhari YouTube channel. So today we are going to discuss lecture number 11, which is regarding polymerase chain reaction, PCR. Most of the you people belonging from lab technologist profession or the belonging from other medical technologies related profession or microbiology profession or biomedical engineering, you people know very well about polymerase chain reaction or PCR. So in this lecture we will discuss in detail what is PCR. Okay, in introduction of PCR we will discuss further, we will discuss different components of PCR, and then procedure means how we can conduct PCR, and then summary or conclusion will be discussed. So first we will go for an intro, means what is PCR and who discovered PCR and what is the purpose of PCR usage, why we use PCR actually. Okay, first introduction of PCR. PCR stands for polymerase chain reaction, as technique used in molecular biology. Okay, it's a molecular biology technique used to amplify a single copy or a few copy of specific segment of a DNA across several orders of magnitude, generating thousands or millions of copies of a particular DNA sequence. Okay, students, first we need to understand what is polymerase chain reaction. Suppose you have extracted DNA, as we discussed in our previous videos, like we are going to extract DNA from bodily samples. Okay, and we have successfully discussed what kind of protocols we can follow and how we can extract DNA from bodily samples. You can see in other videos in my YouTube channel. Okay, so once you have extracted the DNA, and then you have went for quantification of the DNA. Okay, and you assessed what is the exact quantity of DNA which exists actually in your sample or what you have obtained or extracted from bodily samples, and you use usually gel electrophoresis or gel dog for the estimation of exact quantity of DNA in your sample. Okay, or from your samples. So in next step we usually want to go for amplification. Okay, in amplification process actually we want to know about, or we want to amplify a specific segment of the DNA. Actually, why we are going to amplify a specific segment of a DNA? Actually, we want to know about a function of a particular gene located on a DNA. If we want to know about a function of a gene located on a specific segment of the DNA, it means that is not easy, because DNA is a very larger molecule and we need to specifically highlight or amplify a specific segment or target segments which we want to study or observe. For this purpose we need to break the DNA first. Okay, once we will break the DNA, or before breaking a DNA, sometimes we need to locate a gene from where we want to break the DNA. Actually, once we will break nearest to the specific or target gene upon that stage of the DNA or upon that strand of the DNA, it will be easy for us to exactly study or understand the functions of a gene of interest or gene of choice, right? So this is the purpose, means we will be amplifying a specific segment of the DNA, then it will be easy for us to find exact sequences, and once we will find the exact sequences, then we can know about the functions of those sequences. Either those sequences are responsible for production of some one protein. Once we will make some sort of interventions, then there will be a mutation. This will confirm like there is a somehow we can say there are some sort of mutations. And so PCR was actually first time developed by Kyrie-Mullis in 1983. In 1983, Kyrie-Mullis actually used Thermus equitis, a microorganism which can tolerate high temperature. Okay, that was used actually for the DNA denaturation, or for the DNA as a DNA polymerase as well. Okay, similarly we should know about, like, if we want to go for the different components of the DNA, what kind of components we actually require for the amplification of the DNA. Okay, for the amplification of DNA we require certain components which are known as PCR components. The first one is actually a DNA template or a target sequence. तो हमें क्या चाहिए होगा, पहले DNA template या target sequence है कि ऐसा DNA जिसमें हमने कोई जीन लोकेट करना है, जिसको एम्पलीफाई करना है, तो हमें पहले किसी पेशेंट का या किसी पार्टिसिपेंट का बॉडीली सैंपल से DNA एक्सट्रैक्ट करना है. DNA एक्सट्रैक्ट करने के बाद हम एक टेम्पलेट DNA लें जिसमें हमने काम करना है, जिसमें से किसी जीन को हमने आइडेंटिफाई करना है. ओके, DNA template is the DNA molecule that contains the DNA region segments to be amplified, जिसको हमने एम्पलीफाई करना है एक्चुअली. फिर हमारे पास DNA polymerase है. अब हमें एक और जो रिक्वायरमेंट है हमारी फॉर द PCR रिएक्शन, दैट इज DNA polymerase. DNA polymerase sequentially adds nucleotides complementary to the template strand. कि जो हमारे पास ये टेम्पलेट स्ट्रैंड है, सपोज यहाँ पे मैं एक ड्रा कर लेता हूँ, लाइक दिस वन इज अ टेम्पलेट स्ट्रैंड ऑफ द DNA. ओके, लाइक दिस. Okay, and we want to add these are the actually nucleotides. Okay, I will show you guys. So these are some of the sequences, nucleotides. If we want to in next step we need to add DNA polymerase. What will DNA polymerase will do? Actually DNA will polymerase will add some nucleotides over here, okay, at the 3' end. Okay, at the 3' end or hydroxyl group DNA, there would be some sort of we can say nucleotides will be added over here, and then we can say amplification of DNA will take place. Okay, in further primers are synthesized DNA strands of about 18 to 25 nucleotides. तो हमने next step में क्या करना है कि primer synthesize करने, and primer should be complementary to the target sequences. जो target sequences हैं उसके complementary मीन उसके साथ bind होने चाहिए. Suppose these are the primers, like these are the primers. Okay, these primers contains about 18 to 25 nucleic acids. These can be considered as sorry, I will just label them. Here you can see P primers. These are primers. These primers are required for the amplification of specific targets sequences of the DNA. 2 primers must be designed. Okay, 2 primers mean one should be forward primer and the second one should be the reverse primer. तो हम जब भी primer डिज़ाइन करते हैं तो forward primer भी डिज़ाइन करते हैं और reverse primer भी. The forward primer is complementary to the 3' hydroxyl end, where the reverse primer is complementary to the 3' end of the sense strand from starting from 5' to 3', and the forward primer will start from 3' to 5'. So actually 3' to 5, if we want to decide what is 3' to 5', actually the phosphate region is actually termed as 5', where the sugar molecule or the sugar region is actually termed as the 3'. Okay, so in DNA, if I will draw for your easiness, like this one is a strand and this one is a strand. Okay, if this one strand is like, let me show you, the top one strand is 3' and 5', so the second strand will be, okay, 5' into 3', sorry, 5' into like 3'. So this is actually hierarchy or binding structure. Further next we are going to discuss another requirement for the DNA for the PCR reaction, that is Dntps. All types of nucleotides are building blocks for new DNA strand and essential for reaction. It includes adenine, guanine, cytosine. Means we need Dntps or nitrogenous bases. Okay, nitrogenous bases are actually building blocks of a DNA molecule, and these bases will be added upon a template strand, and then there will be these actually bases will fulfill the gap which is created due to, like over here I will show you. This is a primer, this is a template strand, these are the primers, these are the primers on template strand, these are, so there is some gap, like I will show you. So let me highlight this one. This is a gap and this is a gap. This gap is actually filled by Dntps. Okay, Dntps will fill gap. These are some bases, these will be bind over here, so that's why we can say a proper amplification of a gene of interest can be made or would be possible. इसके अलावा हम लोग क्या करते हैं कि मैग्नीशियम विल आल्सो बी एडेड. मैग्नीशियम भी एक रिक्वायरमेंट होती है फॉर द पीसीआर रिएक्शन. मैग्नीशियम क्या करता है कि ये लाइगेशन या अनीलिंग की एक्टिविटी को क्या करता है कि सपोर्ट करता है. जो डीनेचर्ड डीएनए होता है बेसिकली उसकी अनीलिंग एक्टिविटी, उसकी जॉइनिंग को यहाँ पे मजबूत करेगा. सो दैट्स वाई इन पीसीआर रिएक्शन वी आल्सो रिक्वायर